Hi and Assalamualaikum. So in this video, us from Group 3 will be presenting our slide on the making of histopathology slide. I am NURUL and I'll be explaining the brief insight of our project in the introduction. So nice. Our group has chosen the mouse as our model for our project. And it is for the extraction of brain tissue for histological examination. So the mouse models have been more important in the study of uncommon human tumors in recent years, especially brain tumors. So the application of biopsy methods in preclinical environment has the potential to give evidence of responsiveness to new chemotherapeutic drugs in treating brain tumor. So traditional histology tumor analysis may provide us a platform for the creation of targeted treatments and customer treatment procedures that improve results while reducing adverse effects. And then next, in general, brain tissue extraction from mice demands meticulous and precise techniques in the histological workflow. So the histopathology workflow starts from specimen logging, and then we have mice dissection, organ extraction, tissue grossing, fixation, which are among the processes. So there is also tissue processing, of course, which involve dehydration, clearing, infiltration, and impregnation. So after tissue processing, there is an embedding process and sectioning process using a microtome, fishing, and mounting technique. So the staining process is of course also required by using the H&E staining, which is the hematoxylin and eosin staining. And there is of course coral slit. And then microscopes like reviewing, labeling, and recording data and reporting is the last essential step in the histopathology workflow.
Hi Dr. Yu, I'm Linor Anif. I will start with the introduction. Dissection is the process where we separate or cutting apart the tissue. For example, we dissect the brain of mice to examine it. Grossing and fixation also the important step in preparing tissue. These are the methods that involve which are dissection of mice, grossing, and fixation. This is the results that we can get from mice dissection, organs extraction, and also grossing of the brain. After the grossing, we can see how the tissue is being fixed. Lastly, you can see figure 1.6 shows overall procedure of those processes. From the results, we can discuss that mice were dissected based on the selected organ, which was the brain. Grossing is an essential part in obtaining an accurate diagnosis. Also, purpose of fixation is to preserve structure as closely as possible compared to the living condition. Also, there are a few precaution steps, for example, wear goggles, gloves, and also the lab coat. Next is tissue processing. Tissue processing refers to the step needed to take an animal or human tissue from fixation to the state where it is entirely infiltrated with suitable histological wax and can be embedded ready for section cutting on microtome. These are the methods of tissue processing. Next is the instrument used, which are automated tissue processor and paraffin thermal unit. The result shows 4 steps of tissue processing, which are dehydration, clearing, infiltration, and also impregnation. From the result, tissue processing is crucial because poorly processed tissue has a negative effect on section cutting and staining. The main objective of processing is to remove water from the tissue section and impregnate it with another medium that will provide support. Also, there are a few precaution steps, such as processing agents like alcohol and xylene must be replaced during the dehydration and clearing stage.
Hi Dr. Yu, I am ALYA and I'm presenting about the embedding process. So embedding process is a process where the tissue sample will be bound over with a medium which is called an embedding medium all over it. The most common embedding medium that we will be using is the paraffin wax. So the methods in embedding process is the tissues were infiltrated using molten paraffin for a certain period of impregnation. The tissues were embedded using an embedding machine, and then last part is the tissue block will be freezed in the cooling machine before performing the tissue sectioning. And then the instrument that will be used is the embedding machine, tissue cassette, and also molder. The result of the embedding as shown as in the image. And then for the discussion, we can know that the embedding will help in hardening the tissue sample. So when the tissue sample is hard enough and has a fixed shape, it will be easier for the sample to be observed under the microscope. So it is very important to do the embedding process. As for the precaution, we need to make sure that the tissue that we chill in the paraffin mold is always on the flat surface. And also, we need to make sure to use one forceps for any changes that we want to make into the paraffin mold. That's all from me. Thank you.
Good day, doctor. I'm Eshika and I'll be explaining about sectioning and also H&E staining. Sectioning is a process comes after tissue has been fixed. Process is also embedded where the tissue block is sectioned into thin section using microtome. And these are the methods where the tissue is sectioned by microtome and also section was picked up onto the slide using fishing method. These are the instrument used, rotary microtome and also tissue flotation water baths. These are the result. The block is put in the holder and sectioned using microtome, forming ribbon, and the ribbon is transferred into water bath. Then fishing method is used to pick up the tissue onto the slide. and the slide is warm overnight. For a discussion, rotary microtomes works as a manually operated cutting device for all types of work with paraffin and hard cutting technology. And this disposable blade was used to cut the block using this microtome. For the precaution steps, make sure that the paraffin is not overly hard, use a sharp knife, and ensure that the microtome is not set too thick. H&E staining. H&E staining is one of the most common tissue stains used in histology. Hematoxylin is a basic dye that stains purple on acidic structures such as the nucleus, while Ellyson is an acidic dye that stains basic components such as cytoplasm in reddish or pink. Mounting involves the placing of cover slip over a sample of tissue on a glass slide. And these are the methods for staining and also mounting using DPX. The result is slide was immersed into pre-staining solutions at time interval and was rinsed with running tap water before hematoxylin staining and stained with hematoxylin and then rinse again before staining with eosin, and then slide was immersed into alcohol followed by xylene, and slide was mounted with DPX and cover slip was placed. Discussion are, pre-staining process include the waxing, removal of styrene, and adding of water. This is because paraffin wax is not miscible with water and is not permeable to dye. For the mounting with DPX, this will adhere the cover slip to the glass slide. For the precaution steps, make sure to always wear gloves while doing staining procedures and do not overstain H&E. That's all from me, thank you.
So next, I will be presenting about the microscope slide viewing. So right after the staining process is done, microscope slide viewing will be done. This step is very important as we were able to diagnose and also observe abnormal conditions that were available on the slides. And then the instrument that will be used is the compound light microscope by using the method as shown above. And then this will be the result of our microscope slide viewing. And then for discussion, we know that microscope can help us in observing and diagnosing any diseases that is contained as it is contain lenses which can help users to look into those samples in more detail in higher magnification compared to naked eyes. And then lastly, for the precaution, we need to always clean the immersion oil lamps and then always turn off the lights whenever we didn't use the microscope, and then properly handle the microscope as all of the components are very fragile. And lastly, make sure that the slides are always being clipped on the stage to prevent the slides from moving. That's all from me. Thank you.
Good day, Doctor. I'm SIVARANI and I'll be presenting about the microscope slide labeling, recording data, and also the reporting. So for an introduction, I would like to say that the histopathology report should be brief without redundant, unnecessary, or duplicated information. Next, we'll go to the methods. So there are 4 methods for this process. And next, we'll go to the results and also the discussion. So as you can see from the results, this is the microscope slide labeling of the results. And also the discussion, as you can see from the results at the yellow arrow, staining was lighter in the infection area compared with that in normal brain tissue due to the presence of vacuolation and mesenchymal edema. So the black arrow shows small deformed nucleus while the green arrow shows cracked cytoblasts. So reporting dissection of the mice to observe the brain is that the scientists will be able to figure and identify the cure or the treatment towards human brain tumor by using the lab rats. Next, we'll go to the precautions. So the polyester labels for slide labels should also be used as it can withstand extreme temperature. So that's all from me. Thank you.
These are the references we use for our final project. So I guess that's all from us. Thank you so much.